Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PUC first and second year. If you are new to my channel, please subscribe, like, share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, today we shall start today's class. In today's class we shall discuss about the first question, the 30th question. As in previous video we have discussed till 29th questions. Here 30th question is identify the correct pair representing the causative agent of typhoid, which means they are asking causative agent of typhoid and confirmatory test for typhoid. Okay, this pair we have to identify. As we know, here first option is Streptococcus pneumonia or Vidal test. Streptococcus pneumonia, it causes disease called pneumonia, not typhoid, and Vidal test, it is a confirmatory test for typhoid, but we can't choose this option because the causative agent is wrong. Salmonella typhi, it is the correct option, so that is correct causative agent which causes typhoid. Anthrone test, it is used to test carbohydrate, not typhoid. Okay, next Salmonella typhi and Vidal test. Salmonella typhi causes typhoid. Vidal test is used for confirmation of typhoid. Plasmodium vivax, it causes malaria. Urinary tract infection test, this is used for urine analysis. Okay, for urine analysis we are using UTI, urinary tract infection test. Plasmodium vivax is used for malaria. Okay, so the correct option is third one, Salmonella typhi, or it is Salmonella typhi. Confirmatory test is Vidal test. So this you will get from the second year portion, page number 146. Okay, next one. The next question which we have here is select the correct sequence for transport of sperm cells in male reproductive system. Okay, in a male reproductive system you have to go through the diagram of male reproductive system, the sequence of transport of sperm cells. Here you have first option seminiferous tubules, retate testes, then vasa efferentia, epididymis, vasa differentia, and ejaculated duct, urethra and urethral meatus. For this you have to go through the diagram from the second year textbooks on page number 43. On page number 43 in your second year textbook, you will find a diagram. In that diagram, you can find what is the sequence of this male reproductive system. So the correct option is itself first one. Second one, they have given seminiferous tubules, vasa efferentia, epididymis, inguinal canal and urethra. Inguinal canal we have not mentioned in the male reproductive system, nor you will find in the diagram. So this inguinal canal, it is nothing but a short passage which extends to inferior part of abdominal wall. Okay, it's a short passage which extends through inferior part of abdominal wall. This inguinal canal is present both in male and female, and this inguinal canal, it is going to join with the external genitalia of male, but it is not used for the transportation of sperms. So this option is not correct. Okay, next testes, epididymis, vasa efferentia, vasa differentia, ejaculated duct, inguinal canal, urethra, urethral meatus. Again inguinal canal, so this one is wrong, and vasa efferentia, then comes vasa differentia, in between there is epididymis. Okay, this one is not correct sequence. Sorry guys, I forgot to mention fourth option. Fourth option is testes, epididymis, vasa efferentia, retate testes, inguinal canal, urethra. This is the sequence, but I have forgot to mention that fourth option, but the correct answer is this one, first one. So if you go through the textbook, you go through the diagram and you will find the correct sequence of this. And in further second year classes I shall discuss about that diagram. There you will get the answer. Okay, question number 32. Phloem in gymnosperms lack, which means which phloem is not present in gymnosperms? Only sieve tubes, only companion cells, or both sieve tube and companion cells, albuminous cells and sieve cells? It is both sieve tubes and companion cells. Both these are not present in gymnosperms. What is present in gymnosperm? Gymnosperm consists of albuminous cells and sieve cells. These are present in gymnosperms. And in xylem, vessels are not present in gymnosperms. Okay, this you will get from first year, page number 88. First year textbook, page number 88. Next you have the next question. The next question is this: consider the following features. Okay, what features they have given? First one is organ system level of organization. Second one is bilateral symmetry. Third is true coelomates with segmentation of the body. So in this, as they have mentioned here, four features they have mentioned. organ system, bilateral, true, coelomate, and a segmentation. Select the correct option of animal groups which possess all the above characteristics. You have to select which of the phylum is having all the 4 characteristics. For this, if you go through your first year textbook, page number 60, you have a clear-cut information of this. On page number 60, they have given about organ system level of organization column, that is table 4.2. In organ system level of organization, there it is very clear from Platyhelminthes, all the phylums, they consist of organ system level of organization, which means from Platyhelminthes we have Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. Okay, if you open your textbooks and search for the answer, it's better. Pause the video and get your textbook and go through it. Okay, so here we don't have a Platyhelminthes. We can find Annelida, okay, Arthropoda, and Mollusca. These all 3 will show organ system level of organization. Arthropoda, Mollusca, Chordata. Okay, they are showing organ system. Annelida, Mollusca, and Chordata. So all these are showing organ system level of organization, but it has to have all these features. One feature is there. Second one, bilateral symmetry. If you go through your textbook, from bilateral symmetry again starts from Platyhelminthes except Echinodermata, all are having bilateral symmetry, which means all these are also having the second feature, bilateral symmetry. True coelomates, which one are having coelom? If you go through your textbook, coelomate starts from Annelida. Okay, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata, which means all these are having coeloms. Okay, segmentation of the body. Segmentation column, if you find, segment is absent in almost all phylum except in Arthropoda, Annelida, and Chordata. So which means when we said organ system is present in all the phylums, bilateral symmetry is present in all the phylums, coelom is also present in all the phylums, then who is going to decide? The segmentation of the body. Now segmentation of the body, as we said, Annelida, Arthropoda, and Chordata. These 3 are having segmented body. So we have all the 4 features here. So Mollusca, it does not have this one, segmentation. Then rest it is having, then these 3 are having the segmentation. So page number 60, it helps you all to recall all those. Now for time being, you are opening the textbooks and viewing, so it will be very easy if you by heart all those phylums. And then if you mention that Porifera is having cellular, then tissue is having only 2 phylums, rest all are having organ system level of organization. Like that, radial symmetry is having nearly 3 phylums, rest all are having bilateral symmetry. If you remember like this, then it will be easy for you all to recall. So as I said, this from page number 60 from first year textbook. Next question. The correct sequence of phases of cell cycle is, okay, question number 34. The correct sequence of phases of cell cycle is, it is as we have drawn cell cycle. One side we have given it as M phase, the other side is interphase. In that you will find G1, S, and G2. So the sequence is G1, S, G2, and M phase. So it is very easy question. So G1, G2 no, G1, S, G2, M, yes, this is correct. S, G1 no, M, G1 no. So correct option is third one. Okay, this you will find from a page number 163 in first year textbook. Right? Okay, we shall go through the next question. Next question, question number 35. In this question number 35, we are going to find which one of the following statements regarding post-fertilization development in flowering plants is incorrect. It is incorrect. They have not asked the correct one. Which one is wrong? They are asking that. Regarding post-fertilization. In post-fertilization, we said zygote and embryogenesis in first chapter. Okay, they are discussing about flowering plants. In flowering plants, after fertilization, zygote will develop into embryo. Yes, this is correct. Central cell develops into endosperm. Yes, this is also correct. Ovules develop into embryo sac. Ovary develops into fruit. Ovary develops into fruit. Yes. Ovules develop into seed, not embryo sac. So this is the incorrect statement, but correct answer. So question number 35, page number 16 from second year textbook. Even from first year also you will get this, and from second chapter from sexual reproduction flowering plants, from there also you can get, but easiest one I have found in page number 16 from second year textbook. Okay, next question, question number 36. Select the incorrect statement. Again, wrong statement. Inbreeding is essential to evolve pure lines in any animal. Then you have inbreeding selects the harmful recessive genes that reduce fertility and productivity. Inbreeding helps in accumulation of superior genes and elimination of undesirable genes. Inbreeding increases homozygosity. Which one is incorrect? Inbreeding is essential to evolve pure lines in any animal. Yes, it is essential to get pure lines. Continuous self-pollination, what we say, that here it is in animals, continuously inbreeding, it leads to the formation of pure lines. Inbreeding selects harmful recessive genes. No, inbreeding exposes harmful recessive genes. Okay, inbreeding eliminates harmful recessive genes. This is incorrect statement. So correct answer. Inbreeding helps in accumulation of superior genes and elimination of undesirable genes. Yes, this is correct. So that we can superior genes can be identified and the desirable animals can be inbreeded. Inbreeding increases homozygosity. When we are talking about pure lines, then homozygosity will be reached. Rest three statements are correct. Only second statement is wrong. So this is the incorrect statement and correct answer for this question. So this you will get from a page number 167 from your second year textbook. Okay. Next one, question number 37. Which of the following statement is incorrect? Again, which one is the wrong statement? Viruses are obligate parasites. Obligate means what? Obligate means which are totally dependent on that particular organism or a plant or something, without which it could not lead a life. Okay, we have many of those parasites which will be living on our body or without our body or somewhere else, but these parasites are those which won't live without that host organism. It requires host organism. If host organism is absent, then this organisms also dies. So viruses are obligate parasites. Yes, obligate parasites. Nowadays you are hearing about corona. So corona could not live outside, okay, outside the human body or outside the living cell. So it requires the living cell, especially a human body. When it enters into a body, it will be activated and it will function. So similarly, viruses are obligate parasites. Yes. Infected constituent in viruses is the protein coat. This one is wrong. In protein coat is not infectious. Okay, protein coat is not causing infection. Protein coat does not cause diseases, nor it is harmful for us. What is infectious is genetic material. Genetic material will be infectious. Okay, what DNA, RNA will be present inside these viruses, that will be infectious, not a protein coat. Okay, so this is the incorrect statement. It must be the correct answer. Prions consist of abnormally folded proteins. Yes. Viroids lack a protein coat. Yes, viroids lack a protein coat. That does not have a protein coat. All three statements are correct. Only this one is incorrect. So this one is the correct answer. So this you will get from first year textbooks, page number 26. Okay. Next question number 38. DNA precipitation out of a mixture of biomolecules can be achieved by treatment with. Okay, we are going to remove DNA from a mixture of biomolecules. As your first year students, we have studied about biomolecules, carbohydrates, proteins, fats, lipids and nucleic acids. From all those we have to remove only DNA. It is achieved by treatment with what? In second year portion you have this, that is by using chilled ethanol. We are using chilled ethanol. It is a very direct question. Methanol at a room temperature, no. Chilled chloroform, no. Isopropanol, no. We are not using any kind of this. Only ethanol helps to get the threads of DNA, okay, clearly visible in the test tube. So chilled ethanol is used for to remove that from mixture of biomolecules. DNA is removed easily, okay, and it is stored. This you will get from biotechnology chapter from second year, and this is page number 201. Okay, next. Which one of the following is not a method of in situ conservation of biodiversity? In situ means inside their habitat, in their own habitat. Ex situ means away from their habitat, away from their natural habitat. In situ we have zoological parks. In situ conservation we have sanctuaries, bird sanctuaries, national parks, all this comes under it. Ex situ conservation, where we are getting those animals or plants away from their natural habitat and growing in some other area, like botanical gardens, like zoo, these comes under ex situ conservation. Okay, so here they have asked which one of the following is not a method of in situ conservation. Wild sanctuary, yes it is. Botanical garden, no. Sacred groves, yes. Biosphere reserves, yes. So all these three are in situ. Botanical garden is ex situ, so it is not a correct, not a method of in situ conservation, so it is the correct answer. So this you will get from your second year textbook, page number 267. Okay, next one. Which of the following immune response is responsible for rejection of kidney graft? The graft is what? Graft is nothing but which we have got from the donor, or a tissue which we have got from the some other's body. Like a kidney patient, when it has been kidney is not functioning properly, doctors will ask for a kidney donor. Okay, kidney donor it might be, or a graft, or a tissue of any part of the other body when it has been placed in some other part of the body, so that we call it as graft. Okay, which of the following immune response, which of the immune responses is responsible for rejection of kidney? Okay, so humoral immune response, inflammatory immune response, cell mediated immune response, autoimmune response. It is cell mediated immune response, because cell is going to identify the new cell which has been entered inside the body. Okay, this cell mediated immune response, it is going to function through T lymphocytes. Okay, T lymphocytes, these T lymphocytes, they directly attack pathogens or graft. Okay, this will directly attack pathogens or graft. Whereas in humoral immune response, what we said, B lymphocytes will produce proteinaceous antibodies. These antibodies will reach to the plasma and they will fight against those pathogens. Okay, inflammatory immune response. Inflammatory immune response is nothing special. Just while an inflammation of any of the bacteria or something when it enters, then the inflammation, what inflammation occurs and how does body immune system will start to react, that we call it as inflammatory immune response. Autoimmune response, when a particular cell of a body is going to respond to the other cell, that is autoimmune response. But according to the question, immune response which is responsible for rejection of kidney graft, that is cell mediated immune response, and these two responses, first one and third one, you have in your second year. Okay, page, you will get this answer from second year textbook, page number 151. Okay, so total 40 questions we have solved till now. So in my future classes I shall proceed it further and try to solve many more questions, and we shall complete this 2019 NEET question paper very soon. Official NEET. Stay safe, stay healthy.